Good afternoon, everyone. On behalf of CIRM and the Scientific Programs team, I would like to welcome everyone to this informational webinar that will focus on our Discovery Foundational Awards. This is the first on a new series of webinars that we intend to provide to applicants as resources to apply to CIRM funding. The information contained in these webinar presentations and the Q&A will be made available through our website, along with the FAQs derived from our interactions with applicants. And we hope this will be a good resource for applicants that want to apply to the Discovery Awards. The goal would be that applicants for this program consult with CIRM staff only when they need to discuss the specifics of their research project and how does that project fit, but not about general questions that we will try to cover during these webinars. So with that said, we have organized today's webinar in two parts. The first one is going to be this overview, which should take about 40 minutes or less, hopefully, if I go fast. And we will go over the mission of CIRM and how things have changed with our strategic plan and the pillars of funding since Prop 14 and the implementation of the new strategic mission. And then the DISC0 Foundational Awards, we will go over the scope and activities, eligibility requirements, the review criteria, the process, the application components, and then common questions and mistakes that we find that could be useful for everybody. My colleague Kelly Shepard will be going over the last two parts of this webinar. And also, I just want to make sure that everybody gets familiarized with the members of our scientific programs team. You will have the contact information at the end, but we are joined by Dr. Kelly Shepard, who's Associate Director of Scientific Programs, Dr. Uta Gris-Sammer, who is a Senior Science Officer, Dr. Maziar Shah Mohammadi, who is a Senior Science Officer, and Candida Toribio, who is a Project Manager, and you might be very familiar if you are submitting questions to us. So with that, CIRM's mission is to accelerate world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and the world. This slide is going to be a little dense in content, and I will be providing a very high-level quick overview of our thinking in terms of implementing the first of the themes in our strategic plan, which is the advanced world-class science. So as many of you are familiar with, over the past nearly 15 years, CIRM has already demonstrated success in achieving our previous mission through the coordination of established model for therapeutic development based in 5 pillar programs. The new strategic plan is designed to leverage CIRM's proven funding model by enhancing, organizing, and interconnecting these same 5 pillar programs: infrastructure, education, discovery, translational, and clinical. And in order to advance its mission, the plan is organized in 3 main strategic themes: advanced world-class science, delivering our real-world solutions, and providing opportunities for all. Today's webinar is framed again within the first strategic theme, which is advancing world-class science, which has as its main goal to leverage a collaborative scientific knowledge to inspire collaborative research. And there are 3 main objectives within this strategic theme. The first one is to develop the next generation of competency hubs. And the first installment of this is the Shared Resource Labs program. And for those who are interested, our team will be presenting this concept at the October 27 board meeting. We have also a presentation coming up to the science subcommittee this Friday. So if you're interested to learn more about the specific contents of this concept, please listen in to these 2 public meetings. The next goal is to build knowledge networks. Knowledge networks refers to a key endeavor that should provide CIRM with the infrastructure for collaborative approaches and that will allow California to maximize the investment in research by democratizing data through collaborative endeavors. Some of you who have applied already to the Discovery program will have appreciated some of the changes that we have already implemented with the first elements. One of those elements, the first phase of this building knowledge network data infrastructure, is the data sharing and management plans, which is aligned with the NIH new data sharing and management policies. And this Dsmp, as we call it, comes also with a data advisory. So when people apply to our programs, we have guidelines that I will show how you can access those guidelines. And then you will also be advised by our team on how to make these data sharing and management plans more fitting to what we are looking for, and also to be able to frame it within the FAIR principles as well. The third one of the, and before I move on, besides the first phase of the data infrastructure is the Dsmp guidelines, implementing Dsmp, the data advisory, and lastly, there is a data coordinating and management center concept that we are going to be bringing up to the board at the beginning of next year, or the end of June 2023. That's when we are expecting to be bringing this concept to the board. So all of this could be the first phase of the data infrastructure. So the third goal is aligning with existing pillars. And importantly, some of the changes that you will see in the new program announcement for the DISC0 foundational program have to do with the current alignment of programs to the new strategy. We are working towards a shift in the way that we think about funding-based research and how it can align with the collaborative nature and consortia participation. This is more focused towards a more disease mechanism directed approach, and it will be reflected in the upcoming concept changes that we will make to these programs. So with that said, this slide shows our main 3 research pillars: discovery, translation, and clinical. The DISC0 Foundation Awards, not to confuse with the DISC0, I had to make the joke because some people get very confused, is a brand new program that fits into our recurring set of opportunities in the discovery area. And I'm going to go over a little bit more in detail in the next slides of what are we talking about in here. Before we go into details of the DISC0 in particular, as we get this question fairly often, I want to highlight the key differences between the 2 discovery programs. The objectives are quite different. For the DISC2 Quest Awards, the goal is for projects to lead to a development candidate that can progress through the pipeline into the different stages of development and towards translation and clinical. In contrast, with the DISC0 Foundation Awards, there is no expectation of progression. The focus of this program is to address basic knowledge gaps in the biology of stem cells and regenerative medicine approaches relevant to human biology and disease. There is no expectation that the DISC0 will progress down the development pipeline. However, and this is relevant to the alignment with other programs that we are developing in the upcoming year, while CIRM is interested in purely foundational basic discovery, CIRM's goal is that these efforts will ultimately, in the longer term, serve as the basis to create new avenues and provide a rigorous foundation for translational and clinical development work. And we will be providing examples of what we are looking for in the next slides. So in terms of a scope, CIRM is looking for projects that will help us solve critical basic knowledge gaps in the biology of stem cells and regenerative medicine approaches relevant to human biology and disease, and to advance stem cell tools. This is a very broad objective, but we are very interested in research as it pertains to the following key areas: advance our understanding of the biology of stem cells, advance the application of genetic research, and genetic research is now, as you know, in scope as per Proposition 14, and this needs to be relevant to human biology or disease and stem cells or regenerative medicine. Advance the development or use of human stem cells as tools for biomedical innovation, and lead to greater applicability of regenerative medicine discoveries to communities representing the full spectrum of diversity. So what are the activities that are in scope for the Discovery Foundational Awards? CIRM is looking for projects that will help us elucidate critical basic knowledge gaps. The DISC0 funding is explicitly for foundational and or mechanistic research projects. Importantly, DISC0 projects should be grounded in human biology and or pathological mechanism of disease. And as I said, and I will repeat it here, while there is no expectation that DISC0 will progress down the development pipeline, we are also interested in that these efforts will, in the longer term, serve as the basis to create new avenues and provide foundation for translational and clinical development work. Prop 14, importantly, delineates a very strong interest and foundation and funding earmark for CNS diseases. And as such, we encourage the submission of proposals focused on increasing our understanding of the fundamental and mechanistic biology of CNS disorders. And also, we are developing an a structure for our neuroscience strategy that will be implemented also June 2023. So there should be, of course, this is always with the approval of our board. There will be a concept that, if approved, will lead to more mechanistic, multidisciplinary type of funding for CNS disorders, but that should be coming next year. And right now we are trying to focus on pilot projects that could lead to those kind of projects. So here are some examples of the different types of activities that are supported through DISC0. This is verbatim from what we have in the program announcement. And this is also where we would be happy to discuss specific examples during our Q&A for all of you who might have questions in terms of what is that whether we are funding something or not. So the first one is basic research into stem cell mechanisms as they relate to human biology. We are also interested in investigating stem cells or the derivatives as tools for discovering and or enabling therapeutic or other innovations. And this links to two future upcoming programs, actually. So this could lead to generation of data that could be added to as preliminary data to other programs that we'll be having later on. One of them is investigating disease mechanisms of CNS or other disorders with the stem cells or their derivatives. This links to the neurostrategy and multidisciplinary collaborative efforts, omics, target biomarker identification, validation that we will be working on later in the upcoming year. The second one would be modeling disease, developing models of human CNS, again, or other diseases with stem cells or their derivatives. And as I just mentioned, we have a shared resource labs program, a concept that is coming to the board October 27, and our colleague Ute Grishammer will be presenting that concept, and this has interconnectedness with that concept and program and RFAs. And the next one would be reverse translational studies related to stem cell or gene therapy-based regenerative medicine therapies. Some other activities that we are going to fund: basic genetic research. Importantly, it cannot be any genetic research. It has to be genetic research pertaining to human stem cells or regenerative medicine. So basic genetic research as it pertains to that. One example could be projects that focus on its application as a tool for discovering or to enable therapies or other innovations, for example, as given the Proposition 14 focus innovations in CNS. Another example of activities is basic research or tool discovery to address bottlenecks in the development of stem cell-based and gene therapies, such as cell tissue targeting, immunogenicity, or toxicity in the development of stem cell-based therapies. Other examples could be development of genome or epigenome editing tools to support regenerative medicine, or also data science or computational approaches to understand genetic risk of disease. And lastly, we are also interested in research or tools related to diversity, equity, and inclusion in science. For example, use of human-induced pluripotent stem cells or omics analysis from diverse populations, and studying diseases or diseases subtypes more frequently experienced by underserved groups. And there might be already some questions in the Q&A. I can see the red thing there, and we will be handling those in a few minutes when we get to the Q&A. Finally, a few more examples of activities include studying primary human cells and tissues as a ground truth for in vitro stem cell models or to improve regenerative medicine approaches. What we mean by this is that a large part of the field has been focused on the differentiation of pluripotent stem cells into mature somatic cells. But how close are we, how close are the cells that we derive in vitro to the cells in normal development is what we could be focusing in this type of activities. Basically, could be studies to better understand primary human cells and tissues, healthy and or diseased, as ground truth for in vitro stem cell models. And then auxiliary research activities that support regenerative medicine, for example, biomarker discovery or target discovery, tools for genome and epigenome editing or imaging. Importantly, all projects need to tie back to human stem cells and regenerative medicine. Now, before I go into eligibility requirements, I would like to point out that DISC0 allowable activities are extremely wide in scope. And as I mentioned earlier, CIRM is implementing collaborative infrastructure. And in order to be successful, the scope and focus of our programs coming up will narrow down. We will discuss this in more depth as we present future programs at future webinars, but want to bring this point to start socializing this concept with all of you stakeholders. And feel free to ask questions during Q&A or during calls if you have very specific questions about that. Also, just as a reminder, this will not be a done deal up until our board approves concepts. So this is how we are thinking in terms of moving forward, but it needs to be approved. Oh yeah, the human stem cell component must be included in the project. Resources for applicants. So before going into eligibility, I would like to provide a couple of reminders about resources to all the potential applicants. We have a communications team that's not on this call, but CIRM is working very closely with our communications team that maintains our website. And we have, if you go to the main page, and you go to for researchers under information for applicants, you will find these two very important resources. The Dsmp, the Data Sharing and Management Plan Guidelines. Just as a comment here, the Dsmp has been piloted with the DISC, the discovery programs. And piloted means that those guidelines will change a little bit in the coming months, and also the templates will be changing because we are learning the lessons from having implemented in the first round of discovery programs. So bear with us, but this is a very useful resource. The second one is the pluripotent stem cell resource that you will find. These are clinically compatible human pluripotent stem cells for therapeutic development and commercialization. So CIRM, our business development team, has engaged with some organizations that are making these clinically compatible cells available to our researchers. So look at which ones are there, because one of the issues that we've encountered, especially with the DISC2 Quest program, is that we had projects that could go up to the IND enabling activities, and then the line wasn't compatible for clinical activities. So you had to start all over. So now we want you to think about this and start with checking out our resources and ask us questions if you have questions. Another resource is our Human iPSL repository. It's managed through Fujifilm Cellular Dynamics. It has 2,600 lines. It has diverse ancestries, standardized consents for iPS derivation and QC testing, pre-negotiated license agreement available for commercial use as well, available soon, the SNP genotyping data on almost the entire collection, and whole genome sequencing on more than 300 lines. And importantly, focused on CNS, there's many representation of CNS disorders in our lines. And these are just the partners that were involved in the development of this repository and the grantees that derive the lines. And this information is also available on our website, and in that Fujifilm IPS products, you can also find it on the right lower side. You can see and we will make these slides available as well. Eligibility requirements. Who and what is eligible to apply to this program is California institutions. What is a California institution? It is defined in our grants administration policy as an organization that employs and pays more than 50% of its employees in California, and that directs and controls the award activities from California. The PI has to be at least 20% committed to working on the project, must not currently have another application pending review or approval under this specific DISC0 funding opportunity, or another application that's substantially similar or has overlapping activities pending review or approval under any other opportunity at CIRM. Projects need to start within 90 days of Independent Citizen Committee board approval. Project generating omics data need a dedicated PA, and we will flesh this out a lot more as we are as we finalize the Dsmps and all the rules. But this will be something that's very important. It also will align with our data collaborative and management center concept, because those folks will have to coordinate with the DCMC, Data Coordinating and Management Center, for data generation and management and sharing. So that's the idea. So we are taking this very seriously. And then for profit, applicants must demonstrate solvency. Review criteria and process. What does the application review process consist of? Once the application is submitted, CIRM staff reviews the applications for eligibility. As per the slide that we just showed, can the application be reviewed? That's the question we ask ourselves, and we focus on those eligibility criteria. If the application is deemed eligible, it will be accepted for review. Merit review is conducted by our grants working group, which consists of a scientific panel and the patient advocates and nurses from the board. Applications will be scored and critiqued by this grants working group, who submits their recommendation to CIRM's governing board for a final decision. And then the CIRM board makes the final decision for funding. So there is one instance, however, and this usually applies to all the discovery program applications, DISC0 and DISC2, in which CIRM conducts the review in 2 stages, starting with positive selection. This happens when the application volume exceeds the grants working group capacity. We have 15 grants working group reviewers. If we get 100 applications, we cannot flow them through that grants working group panel. So we need to reduce that number. Therefore, when that happens, we have 2 stages of the review. The review process for these high volume programs, the first stage could be the grants working group members conduct a pre-review of all the applications to identify a subset that they believe are most impactful and responsive. For that, there is a part of the application, and that Kelly will show us in a minute, That's very important for them, for you to pay attention to, because that's what they look mostly at when they do the positive selection. Failure to be selected from the positive selection doesn't necessarily imply that an application lacks merit. One can only infer that it was not in any given grants working group members top percentile for funding. The chances of an application being selected can vary depending on the total number of applications that we have received, the subject area of those other applications, and other factors that the grants working group may consider, which could be scientific or other. So if you have questions, you can always come back to us. But again, it doesn't mean that the application was a bad application. It's just that we have too many and you can come in for the next round. That's the good thing. We have 2 rounds of DISC0 and 2 rounds of DISC2 every year. This might change, by the way, but for now, there are 2 DISC2s and 2 DISC0s every year. How do reviewers evaluate merit, and what will reviewers look for in the applications? So they look at whether the project holds the necessary significance and potential for impact. In other words, what value does it offer and whether it is worth doing. Especially, this is especially important in those reviews that have a 2-stage review, because this could be evaluated in that page that I mentioned earlier on. Is the rationale sound? Basically, does it make sense to do this? Is the project well planned and designed? Is the project feasible? Can they do it? Feasibility for DISC0 does include some strong preliminary data. And does the project uphold the principles of diversity, equity, and inclusion? Now I am going to leave the ground for Kelly. Kelly, if you would like to take cover, I'll just let me know when you want me to move forward.
Okay, I'm just going to briefly go over some of the application components to make all of you aware. I know a number of you have actually submitted applications to CIRM in the past for our other discovery stage or even other opportunities, and this will be familiar to you. But this might be especially helpful for those of you who have never applied to a CIRM grant before. So if you could just go to my next slide, Rosa, thank you. So something that you need to pay attention to is there are 2 main sections to any CIRM application. There's a section that you're going to complete online, and that's going to be where you're going to be providing basic details about the personnel involved in the grant, the title, the abstract, publicly available information such as the Statement of Benefit to California, some basic information about subject matter and keywords, and importantly, the positive selection preview page, which is for the first step of review that Rosa just described. Very important. The second major component of the application include a series of uploaded attachments that you work on offline and then attach prior to submitting the application to CIRM. Next, Rosa. So a number of these come with templates that are required. Most importantly is the proposal template. This is the section that's described in the program announcement that includes directions on how to prepare your experimental plan, the rationale and significance, your references, where you describe your team and your resources. This is a Word document. There are page limits and fonts and margins all described in that proposal. So they're not described in the program announcement, but they are described in this template that you can download from the application website. There is also a template that you must use for the data sharing and management plan. There's a template for you to provide your required other support document. There's a template for you to provide biological sketches for key personnel. And for for-profit applicants, there are a couple of additional uploads that are required, which include the financial feasibility and solvency statements. And I'm going to go over a little bit more detail about some of these on my following slides. Finally, in addition to the required uploads, there are some slots where you can provide some optional uploads, including things like letters of support. And if you have it, any information that might be important for review about intellectual property licenses or material transfer agreements. Next slide, please. So when you log into the CIRM grants portal and open an application, It's basically going to look like this. You'll see a series of all the sections that need to be completed on the left with a status indicator. The majority of those that you see listed here are sections that you will complete online. As I mentioned, personnel, the budget. And down below, next, Rosa, there's the upload section. This is where you're going to find those required templates that I just talked about. So make sure when you open an application, one of the first things you do is visit that uploads link and get a copy of any of the templates that you're going to need to be working on. Next slide, please. These are examples of what the templates look like. Most importantly, of course, is the proposal where your figures, your rationale, experimental plans, et cetera, are going to be, the data sharing and management plan, the biosketch, and the other support. I'm just iterating this, how important these templates are, because one common mistake that new applicants make is they will use their own proposal or submit a NIH proposal, and that will get you deemed ineligible right off. So make sure you're using the correct and most up-to-date proposals or templates. And finally, for the for-profit applicants, these are what the additional uploads that you are required to provide. There's a Word document that is the financial feasibility and assessment form, and there's an Excel spreadsheet for the financial solvency. Okay, next slide, please. So again, just to reiterate, do use the required templates, especially for the proposal. Adhere to the prescribed page limits. These are preset for you in terms of formatting, margins, and font size. Do not alter those. Just simply follow the instructions for each section. And once this is completed, you will convert it into a PDF when you're finished, and it will be uploaded as an attachment for when you submit your proposal. Next page, please. Since this is now the second offering of DISC0, I just wanted to talk a little bit about the resubmission process for those of you who are interested in resubmitting an application from the previous round. Now, if your application did not pass positive selection, this means you had no score and no feedback to act on. So you would just treat this as a new application and prepare a new template and just proceed as you did in the last round. However, if you did have grants working group feedback from the previous review and you received a score that you wanted to address this feedback, you would designate your application as a resubmission by selecting the checkbox in the information for review section. That's one of those sections that's online that I showed you in my earlier slide, listed down along the left of your screen. And then in the uploaded proposal template, near the very beginning, there's something called a resubmission statement. This is a page where you're allowed to describe exactly how you addressed the critique and feedback from the previous review. So these are just screenshots showing you where the checkbox is that you'll be looking at if you're submitting a resubmission. Okay, next slide, please. Okay, so now that I've gone over the application components, I'm just going to go briefly over a few common mistakes that we see with applicants each time, and hopefully to help some of you avoid some of them if this might be new to you. So next slide, please. Okay, these are some common issues that are affecting eligibility. So these are the kind of mistakes that get your application not even reviewed at all. Projects that are focusing on animal rather than human biology, or missing key human cell validation studies. As Rosa described earlier, this is a program that is focused and grounded in human biology. Okay. This is also a program that's geared towards addressing knowledge gaps. So you're asked when you put together a proposal to define a knowledge gap and describe how you're going to address it. So even though you might be proposing activities that are allowable, if they're not directed and framed around filling a knowledge gap, that could create an issue around eligibility. Another common mistake is submitting an application that is too mature or is too focused on product development, something that would be appropriate for a DISC2 trans stage or clinical stage programs. One I highlighted earlier is not using the required and appropriate templates for the required sections. In the case of DISC0, if you're proposing a project that includes omics, you need to assign a data project manager who will commit at least 15% effort. And then if an application is proposing less than 20% effort for a PI, that will be flagged. Next slide, please. So those were some issues that affected eligibility. On this slide are some issues that affect merit. So being eligible is enough to get your application reviewed by the grants working group. However, getting funded depends on how they assess the merit. And they use the review criteria that Rosa went over earlier. However, having heard a lot of reviews, there are certain issues that come up that reduce enthusiasm of reviewers. And these are some of the ones that we've heard over time that we thought we would communicate in a general way that might help you think about things as you're putting together a proposal. So projects that appear to be too preliminary or too exploratory, not having enough preliminary data to support the rationale or the likelihood of impact in the duration of the grant. So in this case, this is a 3-year grant. So enough preliminary data to convince the reviewers that in 3 years, this will make a significant impact. Sometimes reviewers view proposals as being too unfocused or not what they quote as CIRM-y enough, meaning there's only a very superficial link to stem cells or regenerative medicine. And this probably doesn't surprise you. There has to be a strong link to stem cells or regenerative medicine in order for something to be eligible for CIRM funding. And a very tenuous one that the reviewers don't believe. If they think this is a proposal that is not really a stem cell project, somebody just threw in stem cells to make it eligible, that tends to reduce their enthusiasm. Sometimes interdependency of aims can be viewed as a weakness in terms of feasibility. So one of the review criteria is feasibility. And if a 3-year project, the key experiment depends on 2 years of building some tools that don't currently exist, sometimes reviewers might see that as more risky than if a proposal came in with the tools ready and able to hit the ground running. So sometimes it's not possible to get around interdependencies of aims, but that's something to think about because sometimes it can be viewed as increasing the risk of feasibility. Sometimes an important expertise appears to be missing from the team. This can be addressed by adding a key collaborator or key personnel or getting a letter of support from somebody. If you feel that there's a significant expertise that would be helpful to have on your team that is missing. A common concern is poor grantsmanship, poorly written, or something that is difficult to follow, and failure to acknowledge relevant existing literature. And lately, reviewers are being asked, and we're putting more emphasis on statements around diversity and equity and inclusion. So it's important to take this section seriously and put your best effort into that. Next slide, please. Okay. That is what I had to say about the common issues and errors. Just a reminder that the applications are due November 8th at 2 p.m., not 5 p.m., but 2 p.m. And if you haven't already created an ID in the CIRM grants application portal, now is the time to do it and the applications are available. And I think, Rosa, that's the end of what I was going to talk about, and I'll hand things back to you before we go to the Q&A.
Yeah, thank you, Kelly. That was great. So this is just the last slide to show who we are again. And we are also hiring a science officer. So if you are interested, check out the employment opportunities. This post is not out yet. So if you are interested, also send us an email to discovery@cirm.ca.gov. With regards to that, we can send you more information if you are. Also, for more information and assistance with this program, you can email the Discovery inbox at CIRM. And importantly, something that we are changing. So some of you are applicants to our translational and clinical program as well. And one of the things that from your experience is that traditionally there's been this model of submission inquiries, and you have a meeting with the team and you can discuss the project and all that. Well, that for Discovery, we get hundreds of applications. It is not sustainable. So one of the things that we are doing is we've created a new process, and this is why we are doing this webinar, so that questions are answerable through the webinar, or the FAQs don't need to go through these one-on-one calls. So I just wanted to make you aware that if you send questions to our Discovery inbox, you will get an email from Candida asking you to fill up a pre-submission inquiry request, and we will then triage whether you need a call or you can have your answer is the answer to your question is within the materials that we have. And with that, I think we can go to Q&A. I think, Kelly, you want to start taking some of the questions. Yeah, I think you will be reading the questions, so let's get started.
Okay. The first question is, are you interested in aging mechanisms as they relate to stem cell aging in humans? Uta, would you like to take that one, or?
Sure.
Sure. Okay.
And actually, I'll combine it with Dr. Rao's second question, as she corrected herself to add also immune cell aging. So with regard to stem cell aging, absolutely, it's an important question that relates or involves stem cells. CIRM is very much interested in that kind of research. If you proposed a project purely about mature immune cell aging, then you might have to be a little bit more creative about thinking about how it fits our requirements for being genetic research related to regenerative medicine, for instance. But yes, CIRM is very much interested in this kind of research.
But also, if you don't mind me adding to this,
Sure.
Uta, I think the immune cell aging is importantly within neurodegenerative diseases and any across, like mechanisms across diseases, is something that's very important. We are going to be issuing what we hope will be a sort of like the disease teams type of programs that we had, but focused on neuro, focused in discovery and mechanisms of disease that cut across different diseases. So if you want to generate some data that's human stem cell related project in the DISC0 that could pilot preliminary data for something that could then become a collaborative type of project with others that cut across diseases, that's something that you should think about because we are very interested. Next.
Okay, next question from Anonymous. What is the historical funding success rate for DISC0? I guess I could take a stab at that. We don't have a very long historical record to draw from. We've only offered DISC0 one time in the history of CIRM, which was a few months ago. From that one experience, I can tell you that the percentage of applications that was funded was about 14–15%. 11 were funded. And you can actually see the public reviews summaries of all 11 that were funded and the remainder that made it through to the second stage of review that were not funded. And you can get an idea of what the reviewers liked and saw as strengths or weaknesses of those applications. And that's on the same website where you can find the program announcement. And I often recommend potential applicants take a look at that because sometimes it can help you figure out how to frame your proposal when you're putting it together, just to have an idea of what reviewers have liked and disliked in the past. Okay. Let's see. Another question is, what happens if a proposal focuses on a research area far outside or beyond the expertise of the review panel? Will external reviewers be invited? Rosa or Uta, would you like to answer that one?
We are, yes, I mean, that's an easy one. We have specialist reviewers. Sorry, Uta, you were gonna, but and one of the things that we are doing as we are shifting and we are aligning the pillars with the strategic plan and the Advanced World-Class Science collaborative endeavors, data infrastructure, we are actually engaging now with many new reviewers that have never reviewed for CIRM. So, you know, if you have ideas of reviewers that you would like, feel free to send us suggestions as well. We have our review team is very active. We have, I believe, a panel of about 300 reviews in our roster. So, but then we also go to specialist reviewers.
Okay, Maziar, I think the next question I'll send your way. Does the $1 million budget cap pertain to the total award amount, or does it refer only to direct project costs?
Thank you, Kelly. So the $1 million budget, that's the maximum allowed for direct project costs. Non-profit applicants may request indirect costs in addition to the direct costs, and all applicants may request facilities costs as well. Therefore, for a project with $1 million direct costs, the total award amount would end up being at around $1.4 to $1.6 million. So please see the CIRM funds calculator in the online application, and this will become clear.
Thank you, Maziar. So the next question from Anonymous is, can a faculty be a PI on one proposal and a co-PI on another proposal? I'll take this one. So the answer is yes. Somebody who is a PI on one application can be non-PI or a key personnel on another applications. CIRM doesn't really have a specific role defined as a co-PI in the sense that a co-PI would manage a separate budget. CIRM applications view all personnel as either a principal investigator or key personnel / collaborators. And so our rules are one PI may submit an application per round, but that person who is a PI on one application is allowed to be a key personnel on a separate application in the same round. Okay. Here's a question I think I'll send to you, Rosa. Does CIRM intend to have 2 rounds of DISC0 in 2023? And will the submission dates be the same as in 2022?
No. So the answer is, I think we are trying to change a little bit this whole philosophy of quantity, and we are trying to think more strategically. So very likely, what we are going to do for next year is that we would have this, we are going to have the Data Collaborative and Management Center concept as well. This is an infrastructure one. We will have just funded the Shared Resource Labs, which is a large infrastructure as well. And then in terms of discovery programs, we are going to hopefully have one DISC0 and one DISC2, and then a very large neuro-focused multidisciplinary type of disease teams type of program. That could be the ideal, but again, this has not been proposed to the board, but this is our thinking right now. And I think that it will also allow for more time for people to think and process things and be more focused on the scope of what we are looking for.
Okay. Next question from Anonymous. I have a question about the need for a data sharing and management plan if omics data are to be generated. If a project is generating only simple RNA-seq data that can be quickly and easily deposited in one of the usual online repositories, I'm confused about how it's efficient to dedicate 15% plus of an FTE to this. Is there any guidance for this sort of situation where there are technically some omics data, but it's very small or trivial piece of the work? Why don't we direct that one to Uta?
All right. Yeah, thanks actually for that question. As Rosa mentioned earlier, you were piloting the data sharing management plans. And this is a very good question that we should keep in mind. So first, I want to say that if indeed your omics data is a very small portion of the data you will be generating, you can certainly make an argument in your application about why you don't need a 15% manager dedicated to that. However, I'd like to take this opportunity also to emphasize that the goal of the CIRM data sharing process is to make sure that the data are shared in a fair, following the FAIR principles of findability, accessibility, interoperability, and reuse. And so what I would like to emphasize is in CIRM staff during pre-funding administration for funded proposals will work with the grantees to make sure that the data sharing does occur in that manner, that good metadata are associated with the data when they are shared in public repositories, et cetera. So I just wanted to give a shout out for the possibility that more time and effort by a data manager may be needed than you might think. But I understand your concern, and you certainly can make an argument for in such cases when omics data are a minor portion of the data generated.
Okay, next I'll take a question from our inbox. And Maziar, if you could answer this one. Can I have an out-of-state collaborator on my DISC0 grant?
Yes, out-of-state collaborators can receive funds from a CIRM award, however, through a subcontract. Of note, CIRM requires that the California organization exercises direction and control over the activities. Please note that the funded California organization must retain any new intellectual property generated from the CIRM funding, and the out-of-state organization with the subcontract cannot retain independent publication rights in any intellectual property, such as invention, technology, or data arising out of the CIRM funded project.
Thank you, Maziar. So I'm going to take a couple of the questions that have come in because they're somewhat similar. One is, can preliminary data involve animal biology work, so long as the proposed studies will all be focusing on human work? And the second related question is, that the project has a lot of preliminary data in mouse embryonic stem cells, but only a couple experiments in human cells, would this be considered weak preliminary data? So yes, preliminary data is preliminary data that supports your rationale or your ability to conduct the experiments that you're proposing. And preliminary data is what supports your project. What we're talking about when we say the project should be grounded in human biology is what you propose to do with your CIRM funding. And it's not to say you can't do any animal work in your proposal, because often testing in models, et cetera, is part of the project. But what we're saying is that the DISC0 project itself should be grounded in human biology. And if it is necessary to do any studies using an animal stem cell or an animal model, there should be some validation to prove that that discovery is relevant to a human system. And if you're working with humanized models or testing a human cell in an animal model, that's considered human biology. Now, to the question of whether it would be considered weak preliminary data, this is really case dependent. It depends on the project. Everyone has up to 3 years to put together a proposal and to state what they think the impact of their findings will be in those 3 years. If you have 0 preliminary data in human system and the whole entire new project is a human and you've never worked with human cells before, reviewers might wonder if you're going to get there in 3 years, right? But if you're well-versed in the techniques, you have experience in the lab, this is routine nature studies for you, then perhaps it could be achievable in 3 years. So the amount of preliminary data, really look at it in supporting the rationale for the project that you're going to do and for demonstrating the capabilities of your team, because those are the 2 components that reviewers are looking at, really. They're looking at the significance and impact of what will be achieved, and they're looking at what will be achieved in 3 years. Can this get done? Is it feasible? Is the right team in place? So that hopefully addresses that question. And let's see. There's another question similar to that. I think I addressed that one already. Does the HIPSC repository have any lines with bulk single-stranded RNA sequencing data already? Uta, do you know the answer to that one?
There is no single or RNA-seq data generally available for any of the lines that's currently associated with it. Now the lines have been used in publications, and so, but at the moment you would have to find that data through publications.
So I see another question about a key collaborator outside of California. Maziar already addressed that question, in that you're allowed to have a contract through a subcontract fund a collaborator outside of California. However, you need to pay attention to the IP rules around CIRM applications. So any CIRM funds that are used to outside of the state, any new IP that is generated from the use of those funds has to stay with the California applicant organization. So often this doesn't matter to the collaborator, but sometimes the collaborator's institution takes issue with this, and in which case it might be important to get some kind of an agreement in place that they will waive any new IP that comes from the CIRM funding if they agree to take it as part of a subcontract.
And Kelly, and the IP rules, do we have it available on our website that they can access it? That is in the...
Yes, this is more about allowable and non-allowable activities, which are defined in the program announcement and the grants administration policy. But IP regulations are also on our website, and we actually have a handout that we can mail, like a FAQ as well. If you send an inquiry to the Discovery email box that was shown on the slide, we can send something, we can send that to you if you're interested.
Yeah.
Okay,
And
let's see.
We should perhaps package all these so that is all this information with the FAQs as well available. But yeah.
Okay, let's see. Anonymous asked, is CIRM interested in funding basic science applications that study diseases that are not represented in the IPSC repository? Apart from CNS studies, are there any other disease priorities?
Now, in terms of Prop 70 or Prop 14, it's very clear we have 1.5 billion mark for CNS diseases. So there is a big push right now for that. Of course, we are interested, you know, the mission is to accelerate the development of therapies. So an advanced world-class science. So that involves all diseases, but obviously there is a very big component of CNS in our priority.
And I'll just add, if you look at the review criteria, there's no specific review criteria that names any specific disease or to put more weight on any given disease. It's all about the significance and impact. So even if you're studying a very rare disease or a disease that is not represented in many other publications or repositories or what have you, if you can describe the unmet need and the significance of being able to make an impact in that area, then that could be rated highly from a reviewer if you make that case. In other words, they're really looking at the potential for impact and the significance. They're not instructed in any way to put weight on any one disease over another.
And Kelly, before we have only 4 minutes left, we don't need to answer all the questions. We will post, so we are going to take all these questions and answer them afterwards in the FAQ. Okay. So let's just choose the most, let's just pick something that's like that could benefit from answering now live.
Okay.
Are the slides available somewhere after the call? Yes, we will make them available. Also the webinar we will make available.
Okay. Somebody asked if we previously submitted a DISC0 that was scored but not funded, but think it may be more suitable as a DISC2 proposal, would that be a new submission or would we mark as a resubmission? First of all, I'm not sure that a DISC0 would make a great DISC2 submission. I would strongly recommend talking to a program officer about this. However, it would not be a resubmission. It would be reviewed as a, there are different things that reviewers are looking at for DISC2 than they're looking at for DISC0. Here's a, let's see. Since basic research can take longer than anticipated, will there be any punishment if we don't meet the timeline? That
I mean, you won't get the money.
First of all, there's no punishment. These are applications, and the reviewers are scientists from out of state who are reviewing the idea and whether or not they think it can be achieved. Once it becomes a grant and is administered, that there's no punishment if the science doesn't work out the way you think it's going to work out. So
well, but there are so for example, for these two, we have milestones and payment. The payments are adhered to reaching the milestones. So in the case of DISC0, we don't have milestones, but we will be looking closely into the research. Now it is research after all. So if you don't achieve it on time, as long as you can justify it to us, we will discuss with you. You will discuss it with your program officer.
Mm-hmm. Okay, so we only have 1 minute left. Should we, did you want to say any closing remarks, Rosa? And we'll just get to the rest of these questions later. There's still quite a few more that have come in.
Yeah, no, the only closing remarks is that we hope this was useful. We welcome any feedback. Feel free to send us a note to our Discovery inbox. We will keep doing this more and more, and we hope that we have more of a dynamic conversation with the stakeholders so we can inform what's coming into our programs as well. So thank you everybody for your participation. Thank you to the team for your contributions to today's webinar. And we hope this is a useful resource to everybody. Have a good afternoon. Bye.
Bye.
Bye, everyone.